Hello everyone, welcome to class 12 biology. In this video, we will discuss about the second process of gene expression, that is translation. In our previous video, we discussed about DNA transcription, and then after the transcription, we saw the details about genetic code. And now we understand that genetic codes are located on mRNA, which the mRNA got from DNA. So the mRNA is copied from DNA through the process of transcription. mRNA carries the codons, which codes for particular proteins. So the amino acid sequence is determined by the sequence of codons present on the mRNA. So we will see the details about how this mRNA gets converted into protein, how protein is synthesized based upon the information present on mRNA. Okay, so that particular process is called as translation. The polymerization of amino acids to form a polypeptide is called as translation. That's the simple definition of translation process. During translation, what happens is there is polymerization of amino acids. We all know that proteins are made up of small monomers known as amino acids. There are about 20 different amino acids which are involved in the formation of protein within biological systems. So amino acids, they are the monomers. They form peptide bonds with each other to form long chains called as polypeptides. And later on, these polypeptides get modified into proteins, right? The functional proteins. So you must have studied that in your class 11 biomolecules, or you might be studying this in your chemistry right now. So the polymerization of amino acids to form a polypeptide is called as translation. But now there is one important information about translation process. The sequence of amino acids, right? The sequence of amino acids is defined by the sequence of bases in the mRNA. So the amino acid sequence, the amino acids, how, in which particular order the amino acids are arranged, the sequence of amino acids is defined, or it's dependent upon the sequence of bases in the mRNA or in the messenger RNA. So I told you before that messenger RNA carries the codons, and the sequence of codons present on the mRNA will be defining the sequence of amino acids in the polypeptide. So that is the important thing to remember. And over here you have the overall representation of translation process going on. So in the translation process, what are all the things required, right? The things required will be mRNA, the first of all, and mRNA is required, right? mRNA was produced during the transcription process. So the mRNA, it binds with ribosome, right? The ribosome is the cell organelle which is present in the cytoplasm of eukaryotes, and it is the membraneless cell organelle, right? It is not covered by any membrane. So this is a cell organelle, ribosome. It is also called as the protein factory. So when the mRNA binds with the ribosome, it starts the synthesis of polypeptide. And in this particular structure, tRNA is the one, or the transfer RNA is the one which brings the amino acids. Individual amino acids will be brought to the protein factory by tRNA, transfer RNA. So these transfer RNA, they bring individual amino acids, and inside this particular ribosome RNA complex, the amino acid will be binded with each other through the peptide bond. So there will be peptide bond formation happening inside over here. And because of that, a long chain of amino acids will be formed, right? And that will be the polypeptide. And when it is released, it will become the functional protein later on. So that is a brief introduction about the translation process. So now we will dive deeper into the translation process. For the formation of peptide bonds between amino acids, energy is required. So therefore, the charging of tRNA, or amino acylation of tRNA, it occurs in the first phase of the translation. So during the translation process, before the translation process begins, before the polymerization of amino acid begins, first what needs to happen is charging of tRNA, or another name is amino acylation of tRNA, should occur first. Why? Because for the formation of peptide bonds, energy is required. So during charging of tRNA or amino acylation of tRNA, what happens is amino acids are activated in the presence of ATP. You know that ATP is the energy currency, adenosine triphosphate. So amino acids are activated in the presence of ATP, and they means the amino acids, they are linked to their cognate tRNAs. So amino acids, they will be first activated in the presence of ATP. So there is an amino acid over here. It will bind with ATP. The amino acid gets charged. It becomes activated. And this activated amino acid will be linked to the tRNA, to their specific tRNA. Right? So I will show you the process over here. So let's say we have a tRNA whose anticodon. Now remember the anticodon, okay? So the tRNA, we have a tRNA over here with the anticodon AUG. So this tRNA, it knows that it has to bind with a particular amino acid, but that amino acid is not charged yet. So what happens is, in the presence of ATP, the amino acid here, the tyrosine, so AUG, the amino acid, the tRNA with anticodon AUG is the cognate tRNA of tyrosine amino acid. So the tyrosine, so I will just show you again, the tyrosine, in the presence of ATP, the tyrosine gets activated, and then the tyrosine is bound to the tRNA, to its cognate tRNA. Okay, and then we get this particular tRNA. So this tRNA is now charged. So this particular process is called as charging of tRNA or amino acylation of tRNA. So that is really important. You have to know this particular process, which occurs in the first phase of the translation process itself. Now, this charged tRNA or amino acylated tRNA, it has got a special property, right? When two such charged tRNA comes close to each other, when they come close to each other, what happens is it facilitates the formation of peptide bonds between the two amino acids. Right? When two charged tRNA carrying two amino acids, when they come close to each other, what happens is the two amino acids will bind to each other through peptide bond. And I told you that for the formation of peptide bond, energy is required. But the energy now energy is present in this amino acid itself, because the amino acids were activated in the presence of adenosine triphosphate. Remember that. Okay, so when they are brought close to each other, what happens? There is a formation of peptide bond between the two amino acids. So that is the significance of amino acylation of tRNA. Now, this particular process can be sped up, or the speed of this particular reaction can be fastened with the presence of a catalyst. So what is that particular catalyst? The catalyst is nothing but the ribosome. Ribosome acts as the catalyst for the formation of peptide bonds between amino acids. So if you recall, ribosome is the cell organelle, right? It's a cell organelle, and it is also called as the protein factory. So what is ribosome made up of? Ribosome is made up of ribosomal RNAs, rRNAs, or they are also called as the structural RNAs. And in association with 80 different proteins, the ribosomal RNAs forms ribosome. So the ribosome is made up of rRNAs and different rRNAs and 80 different proteins. So in the cell, when the cell is not active, if gene expression is not happening, if there is no transcription going on, what happens is the ribosomes, they are present in the form of two subunits, a large subunit and a small subunit. They are separated, right? Two separate subunits, a large subunit and a small subunit. It occurs in this particular situation when there is no translation going on, when there is no protein production going on. But when the protein production needs to happen, when the mRNA comes over here, they will bind with each other. Right, these two subunits, the large subunits and the small subunits, they will bind with each other to form a functional ribosome. And this ribosome will start synthesizing the protein. Okay. So in this particular ribosome, let's see, you have a large subunit and a small subunit. They will bind with each other. Now we have a large subunit here and the small subunit here. In this particular ribosome, we need to know there are certain sites, important sites for the translation process to occur. First important site will be mRNA binding site. The messenger RNA, it has to bind with the site, mRNA binding site present on the small ribosomal subunit. On the small ribosomal subunit, we have the mRNA binding site. In the large ribosomal subunit, we have got these 3 sites. In your textbook, it is mentioned that there are 2 sites. In other books or in other reference books, you will find that there are 3 sites. But the concept remains same. Okay, so there are 3 sites in which A site is there, A site is there, P site is there, and E site is there. These are the sites at which the tRNAs or the transfer RNAs will come and bind with the messenger RNA over here. So the messenger RNA comes and binds over here, and then based upon the codons, the tRNAs will come and bind in the P and A site. And when the 2 tRNAs come close to each other, there is formation of peptide bond between the amino acids, which is brought by the tRNAs. And after the formation of peptide bonds, the tRNA, one of the tRNA becomes free of the amino acid, and then it will exit the protein factory or the ribosome through the E site. So that is the significance of the structure of ribosome and its binding sites. So we see that ribosomes, they are like the enzymes which is necessary, or the catalyst which is necessary for speeding up the process of polymerization of amino acids. Right? In transcription, we have RNA polymerase. In replication, we have got DNA polymerase. In translation process, we have got ribosomes. So ribosomes, they act like catalyst, like biocatalyst, like enzymes, and they will speed up the process of polymerization of amino acids. So now the next thing required will be mRNA. So mRNA, we all know that it was produced during the transcription process. mRNA was copied from DNA. Okay, so in mRNA, a translational unit is there. So mRNA is a long chain of nucleotides. So in this long chain, we have a translational unit, right? And this translational unit is flanked by, right? Flanked means it is from at the right and at the left side. We have got at the right side we have the start codon. So we know that AUG is the start codon, and at the other side we have stop codon. So we have a start codon and a stop codon, and in between we have the translational unit. Now, in the translational unit, this entire thing is they are the codes for a polypeptide. So these are all codons, GGA, CCU, UUC, all these are codons, and they code for specific amino acids. All right. So, but there are other regions which are not translated, and these regions are called as UTRs, or untranslated regions. So untranslated regions are present at the 5' side as well as at the 3' side. So we have got untranslated regions over here. So this is a translational unit. So this is the translational unit from AUG to UAG. This is the translational unit. But those part of the mRNA which is not translated, they are called as untranslated regions, and these untranslated regions are present at the 5' side as well as at the 3' side. I will show you another picture over here. This picture I downloaded from Google. In this one, you can see that from here to here, this is the translational unit. Okay, the mRNA translational unit. But the ones which is highlighted over here, this is the 5' UTR, and this is the 3' UTR. Okay, so these are the untranslated regions. They do not code for any particular protein, but they are necessary. They should be there. Okay, for the translation process to happen. And we know that at the 5' we have the G cap, okay, the methyl guanosine phosphate, and then here we have poly-A tail. So they are also not translated. So these regions are called as untranslated regions. We have now seen the mRNA, we have seen the ribosomes, we have seen the activated tRNAs as well. So now we have seen all the components which are required for the translation process to begin. Okay, so now what needs to happen is the mRNA, it needs to bind with the small ribosomal subunit. So mRNA, it binds to the mRNA binding site present on the small ribosomal subunit. It binds to the small ribosomal subunit at the mRNA binding site. And it binds in such a way that the initiator codon, the initiation codon or the initiator codon AUG, it is present at the P site. Okay, at the P site AUG is present, and at the A site the subsequent codon will be there, GGA is there. Okay, so the next codon is present on the A side, the first codon is present at the P side, the first codon is AUG, which is also the initiator codon. And this particular process, the binding of mRNA to the small ribosomal subunit, it initiates the translational process, and this is called as the initiation. Now in the next step, what happens is we have AUG over here at the P side, and at the A side we have GGA. And at this AUG is the codon, and the codon is read by anticodon present on the tRNA. So we have the tRNAs over here. So we have these tRNAs over here, and these tRNAs, they have got anticodons over here. And based upon codon-anticodon complementarity, the tRNAs will come and bind over here. So we see over here that AUG is the first codon, and the base complementary or the anticodon which is present over here is the complementary base over here, right? The tRNA which is complementary. This tRNA will come and bind to the AUG, and it brings along with it the activated methionine. And at the GGA, this particular tRNA which has got anticodon CCU will come and bind, and it will bring the glycine, activated glycine amino acid. And when these two come in close proximity to each other, when they come close to each other, what happens? The methionine and the glycine will form a peptide bond between them with the help of ribosome. And after that, the ribosome will move one codon at a time. Ribosome now moves one codon at a time. Now at the A side we have CCU, and over here what happens is the next tRNA will come and bind, and there will be formation of again a peptide bond. And after that, when this one moves ahead, this tRNA which brought methionine will detach and will leave or exit from the E side of the ribosome. So this particular process is called as elongation. And during elongation, what happens? The tRNA with amino acids sequentially binds to the appropriate codon in the mRNA. So that is the elongation process. So what happens? So in this particular picture, the picture which is given in your textbook, we can clearly see that on the mRNA, the ribosome, it moves one codon at a time, and when it moves one codon at a time, the complementary tRNA or the specific tRNA will come and bind at the codon by recognizing the codon through its anticodon, and it will bring amino acids, and there is formation of peptide bonds with each other. And after the formation of peptide bonds, the tRNA will exit, the free tRNA will exit from the ribosomes. And this particular process will result in the formation of long chain of amino acids or polypeptides. So this growing chain of polypeptides is called as elongation process. All right. So now after elongation, when the ribosome moves over the mRNA, and when it reaches the end part, when it reaches the stop codon, UAG is a stop codon, you know that, right? So when it reaches the stop codon, a factor, right, a terminating factor will come. It will bind with the ribosome over here, and then the entire polypeptide will be detached, or the entire polypeptide will be become free. It will become free. It will leave the ribosome. Okay, so this is called as termination. So during termination, what happens is a release factor or the termination factor binds to the stop codon. It will bind to the stop codon, and what happens? The polypeptide which is formed over here will be released from the ribosome. That is termination. Now in the actual translation process, we have got initiation, elongation, and termination. So these are the three processes present in the actual translational process. Before translational process, we have amino acylation of tRNA or activation of tRNA. Then we have got binding of two ribosomal subunits, and then after that, the initiation, elongation, and termination happens, and that will result in the formation of polypeptide. Now, before I show you one animation about the translational animation video about the translational process, I want you to draw your attention over here, the differences between translation in prokaryotes versus the eukaryotes. If you look at a prokaryotic cell and a eukaryotic cell, in prokaryotes we don't have a proper nucleus. We don't have nuclear membrane. So all the processes will happen in the cytoplasm itself. The DNA replication will happen in the cytoplasm, transcription will happen in the cytoplasm, as well as translation will also happen in the cytoplasm. Right. So as the DNA gets converted into mRNA, along with mRNA, along with the transcription process, translation will begin. So that means transcription and translation will happen simultaneously. They will both happen at the same time because the mRNA is present in the cytoplasm itself. When it is formed on the DNA, it is already present in the cytoplasm, and the ribosomes will come and bind and it will initiate the translational process. So transcription and translation process happens at the same time in prokaryotes because there is no nucleus. But in case of eukaryotes, what happens is the transcription process happens within the nucleus, right? And since inside the nucleus there are no ribosomes, the translation process will happen when the mRNA moves out into the cytoplasm through nuclear pores. When the mRNA moves out into the cytoplasm, only then the translation will occur, right? So over here, both transcription and translation happens at the same time. Whereas in the eukaryotes, the transcription happens within the nucleus, translation happens outside the nucleus or in the cytoplasm where the ribosomes are there. And if you look at the structure over here, it is not necessary that for each mRNA there is one ribosome. Many ribosomes will bind on the mRNA and start producing amino acids based upon the codon sequence. So one mRNA can produce lots and lots of proteins in a small interval of time, or in a small time frame. So that is the difference between the translation process in prokaryotes and translation process in eukaryotes. So that's the difference. So now we will watch one animation video about the translation process. I already told you these animation videos will help you in your better understanding. Right now you have been imagining when I told you all these processes. Now you can see the animation video and it will help you in better understanding of the process itself.
The genetic information stored in DNA is transcribed into messenger RNA and then translated into protein. Translation begins when messenger RNA binds to the ribosome. The initial transfer RNA occupies the P site on the ribosome. Subsequent transfer RNAs with bound amino acids first enter the ribosome at the A site. The complementary matching of 3 nucleotides on the transfer RNA, called the anticodon, and 3 nucleotides on the messenger RNA, called the codon, ensures the correct sequence of amino acids. The messenger RNA passes along the ribosome in short spurts of 3 nucleotides at a time. As this occurs, the initial transfer RNA is moved to the E site and its amino acid is transferred to the second amino acid at the P site. At the same time, a new codon is presented at the A site. The initiating transfer RNA, which now no longer carries an amino acid, leaves the E site and the next transfer RNA with a complementary anticodon enters the A site. Each time a new codon sequence moves into the A site, a new transfer RNA brings in an amino acid. The old transfer RNA paired with the previous codon is passed to the P site and then to the E site as the amino acid it carried is transferred to the growing amino acid chain. As the ribosome proceeds down the messenger RNA, a stop codon is finally encountered. At this point, the ribosomal complex falls apart and the protein is released into the cell.
So I hope that video was helpful for you. In our next video, we will discuss about the regulation of gene expression. So before you watch the next video, I request you again that you read about this regulation of gene expression section in your textbook and then only you can watch the video. Thank you.